So DNA codes for proteins, and proteins are really important in our cells. They do a lot of different jobs in different parts of our body. So some examples of things that proteins do. In our digestive system, proteins help to break down our food. They are enzymes which help chemical reactions take place. So chemical reactions are what break down our food. In our muscles, the way that our muscles work are proteins that are pulling against each other to cause our actual muscle cells to contract. You've got proteins in your skin that give it the structure so that it's not all wrinkly and saggy like your grandma's. You've got proteins in your hair that make your hair, that give your hair its structure so that it's not all brittle and breaking apart, hopefully most of the time. So you've got proteins all over your body that that do all sorts of important things. Um, Another example of a protein is also in your skin. It's melanin, and it gives your skin the color that it is. So someone like me, I don't have a whole lot of melanin because I've got very white skin. I also have blue eyes, and so melanin gives both your skin and your eyes their color. And blue eyes are, they're just the result of not having melanin in your eyes. So these are all proteins that your cells need to make. And DNA is what codes for those proteins. So by the end of this PowerPoint, you should be able to explain the relationship between genes and proteins, and really explain how you go from a gene to making these proteins in your body. So the instructions for making proteins are coded in the DNA. And proteins are made of chains of amino acids. So amino acids are these little red dots, and this whole thing, this whole chain, and the way that it folds, this is actually the protein. There are some proteins where you've got many of these chains that are kind of wound around each other to make the actual protein. But each individual piece of the chain is an amino acid. And so there's two main steps in protein synthesis. The first one is called transcription, and that's where a section of DNA is coded into mRNA, which can't leave the nucleus. So it's kind of like if you were locked inside of, oh, I don't know, let's say the library, and you needed to get a message out to someone outside. Like, let's say you needed to get a message out to your mom to come and pick you up, but you can't leave the library. Well, you need to send some sort of message out. Most of you would probably send a text or a call to your mom in order to get that message from the library out to your mom who's outside of the library. So it's sort of the same idea, that the DNA can't leave the nucleus, but the message that's coded for in DNA has to get out to the ribosome where the proteins are actually made. And so mRNA is a molecule that transports that message out to the ribosome. And translation is what happens at the ribosome. So it's where the mRNA brings the message and decodes it and actually makes the protein. So since DNA can't leave the nucleus, mRNA takes the message from ribosomes to the nucleus. And just kind of to review from what you read about, mRNA or RNA just in general is similar to DNA except for a couple of things. So they have different sugars. DNA has deoxyribose, RNA has ribose. DNA is double-stranded and RNA only has one single strand with one backbone and one length of bases. And then their bases are slightly different because in DNA you've got T's and in RNA you've got U's, like uracil. And then there's just one type of DNA, and there's three different types of RNA. There's mRNA, tRNA, and rRNA, and they all have special jobs within making proteins. So RNA and DNA are similar because they both can carry genetic information, but they do have those 4 differences between them. So transcription is the first step of protein synthesis, and transcription is basically making the mRNA molecule. And making mRNA is really important because mRNA is going to take the message from DNA out to the ribosome. So mRNA can move in and out of the nucleus, no problem. DNA cannot leave the nucleus. So the process of transcription is actually very similar to DNA replication. So you'll remember that in DNA replication, we had 2 strands of DNA split, and then there was a molecule called RNA polymerase that went along and added new nucleotides. Well, here it's the same idea. DNA does split, but instead of DNA polymerase adding DNA bases, it's RNA polymerase, and that RNA polymerase is adding complementary RNA bases. So here you can see the DNA is in blue and the RNA is in red, and so it's this big kind of bubble looking thing. This is a enzyme which itself is actually a protein, and it creates this mRNA strand, or this red strand, that's able to leave the nucleus. So mRNA is sometimes called the genetic code because it's what's actually getting decoded to make the protein when it leaves the nucleus and goes to the ribosome. So the mRNA gets read in groups of 3 bases called codons. So one codon is made up of 3 bases. So this first codon, AUG, is one whole codon. And that's just the way that RNA, or that the genetic code, gets read. And so each codon then stands for 1 amino acid. So codon 1 stands for 1 amino acid, the second one stands for the second amino acid, and when you put all of those amino acids together, that amino acid chain can then fold up into a protein. Or it might take a couple of amino acid chains, depending on the protein, that all sort of wind around each other to become the protein. But a strand of amino acids can be a protein. So let's try an example. So the first thing that you do to figure out what amino acids a certain DNA sequence codes for is you look at the DNA code itself, and the first thing you want to do is transcribe the DNA into complementary RNA. So you would look at what sort of RNA bases would pair to this DNA. So A's bind to U's, T's bind to A's, G's bind to C's, and C's bind to G's. So it's the same idea as DNA when you're doing like a complementary strand of DNA, except wherever you're tempted in mRNA to make a T, you're actually going to just change that to a U. So A binds to U in mRNA, and T binds to A. So what I like to do then is just go back and draw a dash, or maybe a line, in between every 3 bases, because that's what makes up a codon. So this is the first codon, the second codon, and the third codon. And this is just a really short one so that I can show you an example. So then you're going to use this diagram over on the side here to figure out what amino acids does that mRNA actually code for. So UAU, that's our first codon. So with this chart, you start out in the center, and you start with the biggest letter, is the first base of the codon. So our first base was U, and then we're going to move further towards the outside. Our second base was A, and then further to the outside still, the last base was U. So the amino acid is tyrosine. The second amino acid, or the second codon, CAU. So we start out in the middle with C, and then we move outwards towards A, and then U, and we see it's histidine. And we'll do the same thing with glycine. G, G, U, glycine. So I've got another example here. So press pause right now and see if you can come up with the amino acid sequence that's coded for by this strand of DNA. And then when you're done, unpause it and you can see if you're correct. So the amino acid sequence that you should have gotten is glutamine, proline, and phenylalanine. So the genetic code, you'll notice in that diagram there were a couple of different codons that stood for the same amino acids. Because there's three letters per codon, and a total of 4 letters. So there's actually 64 combinations of different bases that make up codons, but there's only 20 amino acids. So we say that the genetic code is redundant because every amino acid essentially has a couple of codons at least that code for that amino acid. We also say the genetic code is universal because regardless of what organism you're looking at, whether you're looking at humans, plants, bacteria, that same chart works for any animal. So within the genetic code, every protein begins with a start codon and it ends with a stop codon. So you'll notice within that big wheel, there is some place that said start and some place that said stop. And so those aren't, start and stop aren't amino acids, they're signals to the mRNA. Here's where we start versus here's where the protein ends. Okay, so we kind of went through transcription, the first step in making the protein. So again, the whole goal of transcription is that we make this mRNA copy, which can then go out to the ribosome and actually make the protein at the ribosome. Translation is actually decoding that message. So the reason you see some of these words or some of these in blue is because I want you to make a chart of the different types of RNA in your notes and just jot down their job as you're going through this PowerPoint. So you can already fill in the job of mRNA. mRNA is what carries the message out of the nucleus to the ribosome. So decoding this message at the ribosome is translation. So transcription happens in the nucleus, translation happens at the ribosome. So as codons move across the ribosome, tRNA brings the correct amino acid. So this picture isn't great because it doesn't show you the ribosome, but this mRNA is sitting on the ribosome, and you'll see a better picture of this later. But this mRNA is sitting on the ribosome, and it's getting read. So when the ribosome sees CUA, it kind of recruits the right tRNA to come along. And this tRNA has bases on one end, and it has an amino acid on the other. And so it adds its amino acid, and then the mRNA slides along to the next codon. And it continues on in this pattern until it reaches a stop codon, and then you've got the full amino acid chain. So our RNA is actually part of the ribosome. It's actually making up the structure of the ribosome, and its job is to create bonds between these amino acids so that it creates a big long chain. So this picture I like because it shows the whole picture. It shows the whole process of making a protein. So we start with DNA in the nucleus. A section of DNA is called a gene, and genes code for different physical traits or traits within a cell that we don't actually see. And transcription then makes an mRNA, which leaves the nucleus. It comes to the ribosome, and so here you can see the mRNA that's actually sitting on the ribosome. And it'll get read in groups of 3. So one codon on mRNA binds to an anticodon on tRNA. So tRNA actually looks like a T, and on one end it's got an anticodon, and on one end it's got an amino acid. So it binds its amino acid, and then it leaves the ribosome.